It is my honor to present today's keynote speaker, Dr. James Allison, Chair of Immunology at MD Anderson Cancer Center in Houston, Texas. Dr. Allison is a true pioneer in cancer immunotherapy and received the 2018 Nobel Prize in Physiology or Medicine, which he shared with Dr. Tezuku Honjo for their discovery of cancer therapy by inhibition of negative immune regulation. Dr. Allison has spent his career forging advances in new cancer immunotherapy strategies, including seminal work leading to the development of ipilimumab, the first immune checkpoint blockade therapy approved by the FDA. His current work aims to improve on today's immune checkpoint blockade therapies and to identify new targets to unleash the immune system to eradicate cancer. We are thrilled to welcome Dr. James Allison.
These are my disclosures. So I'm happy to have a chance to talk to you at the symposium on immune checkpoint blockade and cancer therapy. What I want to do in the next 20 to 30 minutes is give you a brief review of the background of it and then description of some of the work that we've been doing lately to make this type of therapy extend to more types of cancer and to be more effective in the kinds of cancer that we've used it on. And most of the work that I'm going to be describing here was done by people in my lab and also in Dr. Padmini Sharma's lab, largely Sumit Subhuti, the prostate cancer work that I'm going to describe. So just to show an example of the early success of checkpoint blockade by blocking CTLA-4, is this story. The next two slides will tell is of a patient who, Sharon Belvin, who was 22 years old, just finished college, just got engaged to get married, just starting on her life, and she was diagnosed with metastatic melanoma. And it turned out her body was riddled with the cancer. She had 31 lung mets. You could see in this thoracic section here, 5 of them, 6 of them actually. And and also a brain met that I'll show you in a moment. But she went through 2 different chemotherapies, high-dose IL-2, putative melanoma tumor cell vaccine, and then irradiation for the brain met, and none of which made a dent at all. And then she went on one of the early trials in 2004, 2005 of anti-CTLA-4. And as you can see here, her lung nodules completely disappeared. And also this brain met, this half-centimeter brain met, which had been refractory to the other therapies, also disappeared. And this is her 12 years after that, with her 2 children that she's had. And she's still doing fine today. I see her regularly. She's become a good friend. And so she's, 14, 15 years out from a single treatment and living a normal life. That's what this kind of therapy can do. And the question arises, where did this therapy come from? Well, it came not from testing chemicals to see which would kill a tumor cell easier than it kills normal cells, or from sequencing the genome and identifying driver mutations and developing inhibitors that would stop them from driving proliferation or whatever. It came from just basic understanding of how the immune system works. And so one of the things that came from our work was in the early 80s, we identified the T cell, the structure of the T cell receptor, which is the molecule on T cells that recognizes antigen. And you've probably got about 50 million different kinds of T cells in your body, each of which has a different receptor like that. And at the time, when we worked on that, it was thought that that was the holy grail, and that was enough to trigger a T cell response. But by the late 80s, we realized that that was not enough. There was a second signal that was required that we showed was mediated by a molecule called CD28 on the T cell surface that bound to molecules called B7, which are really found only on a very particular type of macrophage of myeloid cells called dendritic cells that can present antigen and alert T cells that something's going on. In any event, we also showed in the late 80s that tumor cells don't have those B7 molecules, so they're invisible to the immune system. And so they could grow, unnoticed by the immune system for a time until they perhaps reach a sufficient size where they pinch off the oxygen supply or can't get enough glucose or whatever for metabolism. And so they die an inflammatory death that causes inflammation. These other cells come in, these dendritic cells come in, pick up tumor pieces, phagocytize them, represent them on their surface. Now, in the context of the CD28 and B7 pathway, and those two signals together tell a T cell start dividing. Because you only have a few of cells with any given specificity, but you need hundreds of thousands of them to actually go out and deal, with the problem, whether it's a virus infection or a tumor or whatever. And so they've got to proliferate like crazy to generate that number of cells. In the first week or so, they do that in a response. They go from a few dozen to hundreds of thousands to maybe even a few million T cells. But then they have to stop. And what happens is these two signals, while turning on this GO program, also turns on this gene called CTLA4 that initiates an off program that tells the T cell, you got to stop this pretty soon, and inhibits co-stimulation and just stops the response. And so when we worked out this biology, I had the idea that if it's a race then from the time belatedly, because it gets a late start, that the immune system gets started if you can generate enough cells to eliminate the tumor mass before CTLA4 turns them off, then you've got a chance. But if not, then the tumor wins. And so I have the idea that if we just take the brakes off by blocking the CTLA4 molecule with an antibody, then we could let the T cells just get unrelenting co-stimulation and keep going as long as they need. And this was an attractive idea for a couple of reasons. One was because we're treating cancer not by treating the cancer cells, we're treating the immune system. And so this ideally, could work against any kind of cancer. It turns out it doesn't, but I can tell you several reasons why in a little while. But theoretically, it could be a universal cancer therapy. And the second thing is because chemotherapy, radiation, other kinds of conventional cancer therapies kill tumor cells, start this priming that they could sustain. If you give the immunotherapy on top of those drugs, you could potentially give them the benefit of an immunotherapy, which means T cells, for example. Once you got them for the rest of your life. And if the tumor comes back, they can reattack it. And also, the immune system is a living, growing thing. And if the tumor cell changes, the immune system can change with that and adapt to it. Unlike, a single drug that you give or a burst of radiation that you give that just deals with the cells that are there when you give it. You can bring to bear the dynamics of the immune system. And so, great idea, but does it work? So this is one of our early experiments with an experimental cancer growing on the backs of mice. You can see the tumor grows. We inject it into a number of mice, and it grows. And at about a month, we have to euthanize the mice because it gets so large. If we inject antibodies that block that second signal, that CD28 signal, the tumor actually grows faster, showing that there is an attempt for the immune system to do something, but it just can't get quite there. But when we injected antibodies to CTLA4 and blocked those negative signalings, the tumors grew for a while and then went away. Just that one molecule of everything that's going on when your immune system meets the tumor, that one molecule, if you can take it out of action, allows your immune system to eliminate the tumor. And more importantly, those mice are immune to re-challenge with that tumor for the rest of their lives. And so when we got this result, I thought it might slow tumors, but I didn't realize it would be so curative. But after this finding, we carried out a lot of experiments in mice with many different kinds of cancer, animal models. But we could cure either by itself with just anti-CTLA4, or with a little bit of help from chemotherapy or a vaccine or something. We didn't find any tumors that we couldn't cure. And so for several years, I worked to try to get a drug company interested in this, and finally, about 1998, interested a company called Metarex, which is a company that had an interesting technology. They had mice that they replaced their antibody encoding genes with human. And so you could immunize these mice and then get out of them fully human antibodies that were ready to go into humans. You can't inject mouse antibodies into people because your own immune system has recognized it as being foreign. But the human antibodies you could. And so the first antibody was first called MDX-010, later named ipilimumab, unfortunately, by the Food and Drug Administration. But it went into clinical trials later on with the assistance of Bristol Myers Squibb, who helped it in the final development. But it was found to give objective responses in many tumor types, including melanoma, prostate cancer, kidney, bladder, ovarian, lung cancer. So as we had predicted, this could be effective against many types of cancer. A new thing appeared when we started treating patients with this, and that is adverse events. We never saw any in mice, and I could give you an explanation for that, but it doesn't really matter. The fact is that patients that receive these antibodies have have colitis, hepatitis, apophysitis, potentially life-threatening inflammatory reactions that at first were very worrisome because they weren't expected, but there are ways of dealing with them with systemic steroids. And you could treat the symptoms and they go away, and then you wean the patient off the steroids and they don't come back. So these are not autoimmune type defects, autoimmune type problems. They're just, immune related, but they're not your immune system attacking yourself. At least nobody's shown that. And then they're generally manageable without compromising the anti-tumor effects. But now that hundreds of thousands of people have been treated with these diseases, we know that there are genuine autoimmunities that appear in the broader population. Very rarely, a few percent. Type 1 diabetes, although many patients might have had subclinical levels of pre-diabetic conditions, and also myocarditis, a T-cell mediated problem with the heart that is troublesome, but we've developed an animal model for that, and they're looking for ways to treat that. In any event, this went into clinical trials. The registration trial was against melanoma, and this is a result of this trial. This is a trial that overall endpoint was overall survival. So the trial took over 4 years, almost 5 years to complete, to get enough survivors to get a statistical certainty that it was the drug that was doing it. And this shows the data. This line, this dotted line here, is the placebo control. Placebo controls are not used very much in cancer research because they're felt to be not appropriate, except with melanoma. Normally you do the standard of care, but there was no standard of care for melanoma because no drug of any type had ever prolonged the survival of melanoma patients in a randomized clinical trial. So there was no approved drug. The median survival after diagnosis of melanoma was about 7 months or so. So half the people had died within 7 months of being diagnosed with the disease, and fewer than 3% were alive at 5 years. So it's a very lethal disease, which is one of the reasons this was chosen for the first test of ipilimumab. Anyway, the curve here with the dashed lines is the one that shows that the anti-CTLA-4, the ipilimumab only control. What you can see is that the median survival was moved over by about 4 months, which would have been sufficient for FDA approval because, as I said, no drug had ever prolonged survival of melanoma patients before. But the really cool thing wasn't that. It was down here. If you look, you see between 2 years and 3 years, the survival curve flattens out and just stays flat for the 4 years, a little bit plus 4 years of this trial, which means very, very, very few patients are dying after 3 years. And anyway, so this was approved by the Food and Drug Administration in 2011, which means that any doctor can now prescribe this drug. But in 2015, there were sufficient number of patients, over 5,000 in this study, of whom been treated in the early trials for whom there was 10 years follow-up. And so what you can see here, again, is this inflection point in about 3 years. The curve flattens out and stays flat. And so 10 years after treatment, the treatment here is 4 doses at 3-week intervals. So 10 years after that, a little over 20% of the patients are still alive, which was a pretty remarkable accomplishment considering the lethality of this disease. But it's not 100%, it's not 50%, it's just a low percentage. And, so that part is a little disappointing. So why is that? Well, it could be, there's some mechanistic reasons. This works during the priming of T cells, as I showed you in the cartoon. So if that's not going on when the antibody's on board the patients, then they're not likely to benefit. But the other one is, of course, more interesting is there may be more of these checkpoints. When my lab identified this in 1993, 94, and Jeff Bluestone's lab at the same time showed that this was a cell intrinsic inhibitory pathway, which none had ever been seen before, which is a T cell's way of saying, turn yourself off. Was basically what we found. The possibility came up that there were more. And about the time our clinical data started coming out, Tasuku Hanjo, with whom I shared the Nobel Prize in 2018, had identified a molecule called PD-1 that he thought played a role in negative selection of T cells. But in any event, he teamed up with Gordon Freeman and Arlene Sharp at the Dana-Farber Cancer Institute and showed that it had 2 ligands, PDL1 and PDL2, and was another checkpoint. And what they further showed was that this is a bit different than CTLA-4 in that when T cells that have specificity for tumor cells that are trying to kill them, recognize them, and make a substance called gamma interferon, that the tumor responds by upregulating this molecule called PDL1, which then binds to PD-1, which is on all activated T cells, and turns them off. And so that's the way they protect themselves. It's an acquired resistance mechanism. And since then, there's data that shows that this actually is a mechanism whereby the developing fetus is protected from maternal immune responses to paternally encoded antigens. It's involved in protecting the embryo from immune onslaught by the mother, and tumors seem to have somehow co-opted it to protect them. But anyway, there's an antibody made to it. It quickly went into clinical trials with very good response rates, 28% or so in melanoma, 18% or so in non-small cell lung cancer, 27% in renal cell cancer. No responses in colorectal cancer, although shortly after this trial, a group at Johns Hopkins showed that there was a subset of colorectal cancer that had a lot of mutations, consequently a lot more things for the immune system to attack that did respond. And also there were no responses to castrate-resistant prostate cancer, whereas, as I mentioned earlier, CTLA-4 did induce responses too. And in a little while, I'll show you why that is. We figured out why that is. In any event, it also became clear pretty soon that patients that didn't respond to ipilimumab or anti-CTLA-4 therapy could subsequently respond to anti-PD-1 therapy with nivolumab, and vice versa. Those who did respond to PD-1 and progressed could then respond to anti-CTLA-4. So these are independent pathways. So where do you go from there? Well, it's kind of obvious. You got 2 things that look like they have different mechanisms and they don't overlap in the resistance. And so you put them together. And so Michael Curran, who was postdoc at the time in my lab when I was at Memorial Sloan Kettering's, now an assistant professor at MD Anderson, did this experiment where he combined different blocking agents to the CTLA-4 or the PD-1 pathway and showed that they're additive at least. So these combinatorial studies in mice, of course, quickly led to human trial of N-acetyl-A4, which you can see here, ipilimumab, again, about a 20% response. But nivolumab, the anti-PD-1 agent, was added to that. And you can see the response here in terms of survival out at about a year was close to 60%. Again, an additive response. And these data have matured now. The patients in a randomized phase 3 trial now are more than 5 years out, and the more than 50% are still alive. And there's no reason why they won't be alive at 10 years. I mean, we may be surprised, but if they're alive with the monotherapy, there's no reason to believe they won't. And so these patients, I think, could be considered cured, 20% with N-acetyl-A4 by itself, a little over 50% in melanoma with the combination, which is quite something when you consider that when we started this, again, the median survival was 7 months, and only 3% of people survived past 5 years. So, but it's not just a melanoma drug anymore. It's extended to a lot of different kinds of cancer. And one of the things that sort of tells us who's got the patients with the cancers that have a good chance of responding is their mutational burden. Because what we know now is that there are mutations that are involved in driving cancer, called drivers, that actually cause the cancer. But as those are accumulating, there's many more called passenger mutations, and tumor cells differ as to how many they have. From down here to up here with melanoma and lung cancer, and esophageal and bladder, that have hundreds to thousands of mutations per cell. In the case of melanoma, these are caused by ultraviolet radiation induced mutations. When you go out and get a bad sunburn, you've got a lot of mutations from that. The others in lung, head and neck, mouth, esophageal, stomach, bladder. These largely associated with smoking, with carcinogens associated with cigarette smoke. And the irony of all this, if you will, is that these high mutational burdens in these types of cancer made them very difficult to treat by conventional methods. Because if you tried to treat with a drug that inhibited a driver mutation, the chances are the tumors had so many mutations, they had a second driver in there somewhere. And you just select out the ones that responded to the ones you treated, and the rest would be resistant. And so, but this made them sitting ducks for immunotherapy, because all of those mutations, if they lead to a change in amino acid in a protein, whether it changes its function or has anything to do with cancer or not, can be detected by the immune system. Not all of them, but there's rules that they have the potential to be. So in that red box, which were some of the most lethal cancers when we started this work, those all respond very well to monotherapy, either PD-1 or CTLA-4. But if you go down to the left of this, where you look at tumors with decreasing totally on average, mutational burdens, you get down here with kidney cancer, and it is very low, where you wouldn't expect a lot of responses, but it responds very well, actually, for reasons that we don't understand. Dr. Sharma, my colleague, actually led a trial that led to approval of checkpoint blockades for treating this kind of cancer. But I just want to make a point, because I'm going to come back to this in a minute. If you think of tumors that don't respond, one of them would be pancreatic cancer, which is right here on this curve, low mutational burden. Well, even lower than that is prostate cancer, which, low mutational burden, responded to CTLA-4, but not to PD-1. It didn't respond that well to CTLA-4. Anyway, we'll come back to that in a minute. But the FDA now has approved, because of a lot of clinical trials that have been going on, CTLA-4, not just for melanoma, as I said, and CTLA-4 was approved 2011, nivolumab 2014, combination 2015, other anti-PD-1 antibodies, et cetera. Anyway, there are approvals in lung cancer, renal cancer, colorectal cancer. It goes down the list. I'm not going to go through all of them. Many, many different kinds of cancer do respond. Not shown on this list is an approval a couple of years ago by the Food and Drug Administration for a cancer, regardless of the tissue of origin, that might have defects in DNA damage repair, and therefore have a lot of mutations that might, result in changed amino acids. And those respond no matter what the kind of cancer. And so you can treat those with immunotherapies, particularly PD-1 antibodies, if they just have that genetic defect. And there are more kinds of cancer that are getting on here. And notably, though, I should point out two things. One is the response rates of these are highly variable. Renal and bladder, for example, it's around 30-40% or so. As I said, combination therapies with melanoma is 50% plus. Others, it's perhaps lower. But yet, in some fraction of patients, there are responses that will last for a decade, or more. We just have to work on getting those higher. And then some kinds of cancer, as you notice, absent from this list are pancreatic cancer and glioblastoma, which are more problematical. And so we've got a lot of work to do. But if I could just now, in the closing time, talk about some of the work that we've been doing at MD Anderson. So Pam Sharma, who's in GU, genitourinary cancer here, have been doing really pioneering studies in bladder and prostate cancer, but way before CTLA-4 was approved for treatment. Trials that she did that were really designed to get tissue and study mechanisms. And this is a very powerful way to try to improve the response rate and learn how to, in a data-based, rational way, apply immunotherapy. What this means is we've already done, the laboratory interrogation, found that, these checkpoints are useful. So they went into clinical trials and they're approved now. But what the philosophy of our new laboratory is that there's an iterative process where you do reverse translation. So you get samples of the tumors from the patients, take them back to the lab, analyze what's going on. If you see something that looks interesting, like a particular phenotype of T cells or a particular phenotype of macrophages or myeloid cells, then you do mouse experiments to see what happens if you could manipulate those. And then you take that back into the clinic again. And it's this iterative process where you go to the clinic, do the treatment, come back, get samples, interrogate them in laboratory, reverse translation, and then do this and do it as fast as possible. So we've done, I think it's now 110 trials, close to 4,000 patients that we've enrolled. A lot of samples that we've analyzed. So I just want to tell you a few stories to show you, what we've learned, to illustrate, what can be done and what I think is the path forward. So cold tumors, cold meaning not very many mutations, as I showed you in prostate, and maybe not much infiltration. So as I told you, there were responders to anti-CTLA-4, to ipilimumab, enough that there was a phase 3 trial done to see if this would be better than the standard of care. And it turned out, in this case, it was placebo. But anyway, there wasn't. There were occasional responses, but statistically, you couldn't show that they were better than no therapy. And so that, with anti-CTLA-4, that failure plus the lack of responders of any responders to PD-1 led Bristol Myers Squibb to totally abandon their prostate program. So let's look at the picture now. So if you've got a tumor like melanoma, and I'll show you some data in a moment, that's fully infiltrated with immune cells, T cells, myeloid cells, others, all you have to do is start thinking about blocking the checkpoints that are there and unleashing the cells. But if you've got a cold tumor that maybe doesn't have many mutations, and also there's not any immune cells in, it's not going to do you any good to give the checkpoint blockers because they've got nothing to work on. There's nothing in the tumor. And so you've got to get the cells in. And so this then just is a comparative study that was done in our immunotherapy platform, showing here pancreatic cancer in terms of of CD8 T cell infiltration versus prostate versus bladder versus melanoma. And you can see there's a lot more in bladder. CD45RZ, which is, T cells that have been experienced, you can see pancreatic, again, is very low, maybe a little bit more in prostate, but a lot more in bladder and in melanoma. And out here, granzyme B, these are CD8s that can kill tumor cells. A lot more in bladder and a lot, even more in melanoma, but relatively few in pancreatic and prostate. So immunologically, then, prostate's a cold tumor. So Dr. Sharma then carried out this pre-surgical trial where she took patients that had localized disease, localized prostate cancer, and gave them a couple of doses of NRC-284 before they went to surgery. Now, the surgery would have been curative, hopefully, for the cancer. So there was not necessarily any therapeutic benefit from getting the epidermal, except that we got the tissue that we could dissect and see what's going on. They also got hormones, but it turns out they didn't really do too much. Anyway, the bottom line is this. What we found was, if you look then by staining, which would be indicated, antibodies would give to these different molecules on the surface of cells would give you a brown color. No CD4s, no CD8s, these are regulatory T cells, no experienced T cells at all. It's ICOS molecule, which is important on a particular kind of cell that can make cytokines that kill tumors. Granzyme B, which is involved in killing. And then macrophages, virtually very cold, very sterile, cold environment. But once the patients have gotten a couple of doses of NRC-284, a robust infiltration with CD4, CD8 cells, get some regulatory cells, a lot of cells here, and macrophages start to come in, and a lot of granzyme B producing cells. So CD4 can drive T cells in. PD-1 does not, apparently. So this then suggested, an answer to the problem, that PD-1 didn't work because there were no T cells in the tumors to work on. And CD4, why didn't it work though? Well, that's because we did gene expression analyses. What you can see is all of these things in this red oval here. These are inhibitory molecules. PD-1 is ligand, PD-L1, VISTA, another inhibitory molecule, LAG3, another inhibitory molecule, which is expressed, receptor, which is expressed on T cells. So CD4 does get the T cells in, but by this compensatory effect, they upregulate these additional negative regulators. And here you can see that with PD-1 on immune cells, but not the tumor, PD-L1 on immune cells and the tumor. And then this other molecule, VISTA, also on cells, immune cells, not tumor cells, but on other cells. And these cells can inhibit tumors. We showed that in some other work that I don't have to talk about. And this is just a numerical representation. After you treat with NRC-284, the CD8 T cells themselves begin to express the ligand for PD-1, as do a lot of macrophages and a lot of the tumor cells. So, we've gotten the T cells in, but it's a suppressive environment. And so we went to Bristol Myers Squibb and showed them these data and said, look, here's the deal. You need to get NRC-284 to get the tumor cells in, and then you can give PD-1 or PD-L1 antibodies to block that pathway. And maybe you get some responses. And if you don't, then you can also come in with anti-VISTA, because we know it's there too. So we did an initial trial, meaning mostly Dr. Sharma, Dr. Subhuti, of patients that got IPI plus Nivo, NRC-284 plus PD-1, basically with the same dose schedule that was given to melanoma patients. And there were some remarkable responses in that. This patient, for example, his PSA was going just sky high, got up to over 400, close to 500. After the first dose of NRC-284, it dropped, and then by the third dose, it had dropped undetectable and stayed there. This patient had a lot of metastases to liver and other soft tissues, and was a complete responder. The PSA become undetectable. The radiographic evidence suggested that, all his metastases were cured. Not bone mets, but I'll come back. Another patient, I'll come back to that. But the problem with this trial was that it was a little too toxic because prostate cancers on average are older and sicker and can't stand the adverse events that CTLA-4 gave. So Dr. Sharma has proposed and is leading several trials that are underway now, multi-institutional trials, combining anti-CTLA-4 in various settings of chemo-naive and also prostate cancer patients that have castrate-resistant prostate cancer that have had chemo, and doing tricks to diminish the CTLA-4 dose by spreading it out in time or giving less overall doses. So these are underway now, and we hope for a positive outcome. And we'll just have to see. But this is how you can go in, and by studying what happens in the tumor, design your next step. Then the next question arises, of course, if the antigens are encoded, by mutated genes, and there's not that many mutated genes in prostate cancer, are there enough to, even if you get the T cells in, are they going to recognize antigens? And so what we did in this trial, which I had a little bit to say about the design of it, but anyway. So we took metastatic castrate-resistant prostate cancer that had resection of their primary tumor. We did genome exome sequencing of the tumor and of their blood cells to determine mutations, and then confirm those that were expressed by sequencing RNA from the tumor. And so we got a good idea about what the tumor mutations were. And then also we know there's 3 shared antigens that prostate cancers have, their tumors have, that I'll come to in a moment. But the patients then went on anti-CTLA-4 therapy, 4 doses that were bled right before each treatment. And then we took their T cells and made all the peptides, made all the neoantigens, all the things that were predicted by the mutations, and looked for whether they could actually detect the products of those. Remember, melanomas have hundreds to thousands per cell, and many of those have shown to be immunogenic. And so I'll just show you this. We're just going to concentrate on one patient here because of time. This one patient, 7, had a very good response. He responded. He's still responding to anti-CTLA-4, and is doing quite well. So when we looked at his genome and RNA sequencing, patient number 7 had a total of 13 non-synonymous, meaning mutations that might have changed an amino acid and a protein, and 10 of those were actually expressed. Only 10 of them. So this patient, again, it's not like melanoma where it was, hundreds, several hundreds, but 10 only. And very surprisingly, when we screened all 10 of those, we found that 2 of them, and it really doesn't matter what they are, but because they don't have anything to do with anything except they just had an amino acid change, so the immune system was alerted to their presence, is nucleotide exchange factor 37, where you can see this is the wild type peptide and the mutant peptide. And this is the assay where you look for gamma interferon production by the T cells from the patient. There were no responses until after the anti-CTLA-4, but then that induced these neoantigen specific T cells, and also a second mutation in a molecule called dihydroxypyrimidinase. Again, a point mutation. T cells didn't detect the wild type, but did respond to the mutant peptide. And remember, this patient was a responder, and he did respond, even though he had 10 expressed mutations. 2 of them the immune system could respond to by making gamma interferon producing T cells, which had the capability of killing tumor cells, presumably. And it'd be I wish I could say that those were the targets that led to the curing, but I can't say that because I mentioned there are shared antigens in prostate cancer. One of them is prostate-specific antigen that is the common blood test that's used to assess tumor burden. And we found patients that had T cells to that, but we found that in both responders and non-responders, so that wasn't particularly helpful. That's a secreted protein, and probably T cells that detected that would not be able to home in on the tumor cells themselves because the protein's probably decorating every cell in the body to some extent. But we found T cells specific for prostatic acid phosphatase and prostatic specific membrane antigen only in responders. So these shared antigens. So we're not sure what the story is, what it is, but at least, patients with very small numbers of mutations can respond, and that together with these shared mutations can lead to a response. And so now we know that there are a multitude of these inhibitory pathways. CTLA-4 was the first, PD-1 the second, and those frankly are probably the major ones of these other ones that I mentioned. VISTA, TIM3, LAG3 play a role. There's a few others that are coming along. They're minor. They may add, though, if we can, figure out how to block them. They may add a few percent of responses. We don't know. There's still a lot of work to be done on them. But the future, I think, of this field is combining blocking our multiple negative checkpoints or giving agonist antibodies to positive checkpoints. We've got a couple of examples of that I don't have time to talk about. And while I don't think that immunotherapy is going to replace chemotherapy or radiation therapy, I think that checkpoint blockade is going to become a part of every kind of therapy, including chemotherapy and radiation therapy. And I say that because, as I mentioned in one of the very first slides, is that this whole immune response is started off by cross-priming. It's induced by killing some tumor cells and causing inflammation. Whether you do it by chemo or radiation, doesn't matter. But what we're going to see is a change in chemo and radiation where the practitioners of those therapies don't try to kill every last tumor cell. Because unless they engage the immune system, they have to kill every last tumor cell or they'll lose. But if we can adapt the dosing, both in terms of amount, but scheduling, to spare the immune system, and I think if we just use chemo and radiation with the idea of obtaining vaccination of the patient with dying tumor cells, then we can move a long way. And radiation, and I include in that X-radiation, which we've done, heat freezing, even focused microsound. And I think that targeted therapies pretty soon we're going to figure out how to use them in combination with immunotherapies if we know that they don't inhibit pathways that are important to T cells. But just to finish up, this is the way that cancer research has been operating since the advent of chemotherapy, and that is that you take a large number of patients, you treat them with an agent, and then you statistically look at whether you've improved the median survival, significantly. And if so, then you've prolonged life, and that's a useful thing to treat patients with to get benefit. But we know from ipilimumab now that you can get the immune system to give you that, but also to give you a tail on the survival curve of about 20%. And the hope, and this is not, the red line is not data, it's aspirational, that our goal now needs to be not so much doing this, but finding the right combination by rationally putting things that work well together to get the durable responses, the tail on the curve, if you will, as high as we can get it. And the diseases that respond now, get melanoma up from 50+% to 85 or 90 if we can, to get bladder and kidney, up from 30 or 40% to more than that. And also, bring this to the kinds of cancer that don't respond now, like very well, like pancreatic and glioblastoma. And the good news is we know the basic rules, and we know that if we just work hard enough, and we learn more about the immune system and how different treatment therapy regimens interact with the immune system, that I think we can do this. And so that needs to be our goal. And so I thank you for your attention and your patience in listening to this talk. I wish you a good evening.